Matabadi. Welcome to our YouTube channel. My name is Komala. I am here with my teammates. We are a team of 4. Today we are here to present you a project on genomic data analysis using PCA and t-SNE. In this project, we aim to analyze genomic data using advanced dimensionality reduction techniques and statistical methods to uncover meaningful patterns and associations. Our objective of the project is to reduce the high dimensionality of genomic dataset using PCA and t-SNE to visualize complex data relationships effectively, to identify significant genetic markers and disease associated variations using statistical tests. We will use various tools and techniques including pandas for data handling, seaborn and matplotlib for visualizations, and machine learning techniques like PCA and t-SNE for dimensionality reduction. Statistical tests like t-test, ANOVA, and chi-square will help us analyze relationships in the data. Our first step is to load the genomic dataset with the use of pandas library. Install pandas. For this purpose, here is a code snippet. We install all the packages needed for the project, and we import the files. Here we are reading a data from the CSV file named IGSR HG01350 undefined.csv. This is the dataset for our project. After loading the dataset, it's important to inspect its structure. We loaded the dataset. To check the data structure, we use the following commands to check the number of rows and columns, the column name, and to view a few rows of the data. Running this code will print the shape of the dataset, which tells us how many rows and columns are in the data. We also see column names to understand what information is stored in each feature. Lastly, df.head displays the first 5 rows of the dataset, giving us a quick snapshot of our data. Handling missing values. We can fill the missing values with the mean of the respective column. Here is how we do that in a code. df.fillNA df.mean of in place is true. This replaces any missing numerical values with the column's average, ensuring that no data points are lost. Dropping missing rows. Alternatively, if we have too many missing values in a certain rows, we can drop those rows entirely using the following code. df.drop in place is equals to 2. Removes the rows with the missing values. Depending on the dataset and analysis requirements, we can choose the most appropriate method. To summarize my part, we have loaded the dataset, explored its structure, and handled the missing values to ensure our data is clean and ready for further analysis. Clean data is essential for accurate insights, especially when working with the genomic dataset. In next section, my team will explain how we process the data, including encoding categorical variables and standardizing the features.
Hello everyone, my name is Gaura. In this section, I will explain how we pre-process the genomic dataset before applying dimensionality reduction. Proper data pre-processing is essential for accurate and meaningful analysis. Our pre-processing steps include encoding categorical data and standardizing numerical features. In this first, we are converting categorical data into numerical features. Why we have to encode this categorical data? In our project, the data sets we selected contains categorical features like it contains analysis group, sample, populations, data collection, data type like this. They are containing a categorical features. So, machine learning algorithms will not work with the categorical features, only work with the numerical values. So, in order to convert these categorical features into numerical data, we are using some functions, encoding functions such as label encoding, mapping, or one hot encoder. In label encoding, we have to assign numerical values to every single input, but in one hot encoder we can independently encode by converting categories into independent binary column. So we are using one hot encoder in this one. Next is converting categorical data to numerical using one hot code. Here df.select is used to identify all the categorical columns in this data sets, and then we are applying one hot encoder which creates a separate binary column for each category. For example, if we have a population column values like A, B and C, the encoder will create separate columns indicating whether a sample belongs to each population. Here we use drop to avoid multicollinearity, which can arise if we encode all categories without dropping one. And in this also we are using fit transform to get the categorical columns and stored it in the categorical encoded, and also we are using data frame to get the categorical columns and store it in categorical encoded df. And next we are printing the categorical encoded df, which will be coming like this output when we run this one. This one is the categorical output. Here one is there 0 0. For every column we are getting this one. This is the output for encoding the categorical into numerical features. Next is combining numerical and encoded categorical data. Once we have encoded the categorical columns, we combine them with the numerical columns to create a single data set for analysis. Like we are getting column wise again and again. For getting in single data sets, we are using this combining numerical and encoded categorical data. In this, we extract all numerical columns and concatenate them with the encoded categorical data. This is concat function, and we are concatenating everything. And this gives us a final data frame that contains only numerical data, which is ready for further analysis. So we can see here output. So we are running this. This is first output. Wait. Here this is the output for this one. This is output. Next is standardizing the data. Why we have to standardize this data? Standardization ensures that all features have a mean of 0 and a standard deviation of 1. This step is crucial because dimensionality reduction techniques like PCA are sensitive to the scale of the data. Here we are using standard scalar as a function to standardize the data and store it in a scalar, and fit transform also to get the final df and store it in scalar data. We create an instance of standard scalar and use the fit transfer method to scale the entire data set. This ensures that all features are on the same scale, preventing any single features from dominating the analysis due to its magnitude. So the output of this standardizing the data is, this is what we get after standardizing the data. This is the output we get here. This is the output, standardized output. So this is what the pre-process techniques will show this one. To summarize, we pre-process the data by encoding categorical variables using one hot encoder, this one hot encoders, and standardizing the entire data set using standard scalar. These steps ensure that our data is clean, consistent, and ready for dimensionality reduction. In the next section, my teammate will explain how we applied PCA and t-SNE to reduce this dimensionality of the dataset and visualize important patterns.
Hello everyone, I'm Akhila, and in this section I will explain how we apply dimensionality reduction techniques like principal component analysis (PCA) and t-distributed stochastic neighbor embedding (t-SNE) to our genomic dataset. Genomic data typically has a very high number of features, making it hard to analyze directly. Dimensionality reduction helps simplify the data while preserving its essential patterns and relationships. So what is PCA? PCA stands for Principal Component Analysis. It is a statistical technique used to reduce the number of dimensions while retaining as much variance as possible. It transforms the data into new axes called principal components that are ordered by the amount of variance they explain. Here is the code we use to apply PCA to our dataset. In this code, we apply PCA to reduce the dimensionality of our data to a maximum of 10 components or fewer, depending on the number of features. The transformed data is stored in a new data frame for easier handling. Explained variance. So the PCA calculates the proportion of variance explained by each component. Here's how we printed the explained variance ratio. This output tells us how much is retained by each principal component. Typically, the first few components captures a majority of the variance. PCA visualization. To visualize the PCA results, we plotted the first two principal components using Seaborn and Matplotlib. Each point in the plot represents a sample from the dataset. This plot helps us identify clusters or patterns in the data after dimensionality reduction. Let's see what is t-SNE now. t-SNE or t-distributed stochastic neighbor embedding is another dimensionality reduction technique. Unlike PCA, which is linear, t-SNE is non-linear and focuses on preserving local relationships in the data, making it more effective for visualizing computer patterns. Now here's the code we used to apply t-SNE to our scale data. In this code, we set the number of components to 2 for easy visualization. The perplexity parameter is set to control how the algorithm balances local and global relationships. We also use a random seed for reproducibility. Finally, we plotted the t-SNE results to visualize clusters in the data. This visualization highlights patterns and relationships in the data that might not be captured by PCA. So this is the PCA visualization and this is a t-SNE projection, as we can see. To summarize, we used PCA and t-SNE to reduce the dimensionality of our genomic dataset. PCA helped us retain the most important information while t-SNE provided a detailed visualization of the data structure. Together, these techniques allow us to explore patterns and clusters in our high dimensional data more effectively. In the next section, my teammate will discuss the statistical tests we apply to analyze relationships in the data and identify significant genetic markers. Stay tuned.
Hello everyone, I am Shekhar. In this section, I will walk you through the statistical tests we applied to our genomic dataset to identify significant patterns and relationships. Statistical analysis help us to discover important markers and variations that may be associated with different populations and conditions. T-test for comparing two groups. A t-test is a statistical test used to compare the means of two groups. It helps us to determine if there is a significant difference between the two groups. Here's how we apply t-test to compare two groups in a dataset. First, we identify the two groups to compare based on a column colored population. We then perform a t-test to calculate p-values for each feature. If the p-value is less than 0.05, it suggests that there is a significant difference between the two groups for that feature. ANOVA for multiple groups. ANOVA or analysis of variance is used to compare the means of more than two groups. It helps us to determine if any of the groups differ significantly from each other. Here's how we applied ANOVA to our dataset. In this code, we group the data by the population column and apply the F one-way function to calculate the p-value for ANOVA. A p-value below zero point 0.05 indicates significant differences between the groups. Chi-square test for categorical association. The chi-square test is used to determine if there is a significant association between two categorical variables. It helps to identify relationships that may not be obvious. Here's how we performed a chi-square test to categorical columns. We create a contingency table using pd.crosstab and apply the chi to contingency function to calculate the p-value. A p-value, low p-value indicates that the categorical variables are associated. To summarize, we applied t-test to compare two groups, ANOVA to analyze difference between multiple groups, and chi-square test to identify association between categorical features. This statistical test helped us uncover significant patterns and variations in our genomic data. Overall, our project combined data processing, dimensionality reduction, and statistical analysis to explore complex genomic datasets. We used PCA and t-SNE for effective visualization and applied statistical tests to gain deeper insights into potential disease association and population differences. Thank you for watching our presentation. We hope you found our project on genomic data analysis insightful.